As I see that most of the participants have joined, so I think I can start. Our dignitary Professor Barshanjit Mazumdar has also joined, so I think I can start. Hello everybody. I welcome you all on the second consecutive day of COVID series of lectures. Today we have Professor Barshanjit Mazumdar with us. Welcome, sir. Professor Mazumdar is a senior professor of molecular genetics in the Department of Biological Sciences at Cleveland State University, USA. He did his PhD from Bose Institute, Kolkata, and did his postdoctoral research at Cleveland Clinic Foundation Research Institute of Ohio, Cleveland. Sir is a brilliant researcher and an educationist as well. His present research focuses on molecular mechanism of inflammatory gene expression in monocytes and macrophages and resolution of inflammation. Also, translational control in host-virus relationship using pathogenic RNA virus as model. Till date, he has been awarded with an extramural research funding of about $5 million, huge amount, for his laboratory. He has 41 scientific publications in renowned international journals and received many prestigious awards for his outstanding research, including RO1 Grant Award from NIH, Grant in Aid Award from American Heart Association, and Faculty Merit Award of Cleveland State University a number of times. We are really feeling honored, sir, to have you today with us as a speaker. I hope we will be enriched and learn a lot from your lecture today. One more thing, participants, we need to join a second time once this session is over using the same link. So I request Professor Mazumdar to present his lecture. Professor Mazumdar. Sir, please unmute yourself.
Can you hear me?
Yes, sir.
Okay. Thank you, and really it's a pleasure to talk with you guys here. I left so this is the first time that I'm entering a college classroom in India. Although I am also a professor, I teach significant teaching we have and interact with students, but I never had the opportunity to enter a classroom in India, in last 30 years. So thank you, Swapna, for arranging this. So, okay. So can everybody hear me well? Okay. So I'm going to start. I'm going to start the screen share, and will tell something about this SARS-CoV-2 virus that is causing the current pandemic. So let's start with the screen share. Okay, so I'm going to talk today on the molecular insight and the potential therapies and vaccines, that what's going on in that front on SARS-CoV-2. So why this virus is causing pandemic? Why it is pandemic? So just look, this is March 1st, 2020. This is the disease death per day worldwide. It's the average of last 5 years. Per day, how many people are dying? March 1st, tuberculosis was the highest. 3,000 people are dying per day. Look, COVID-19, only 56 people. Now, let's look at March 28. COVID-19, 3,233 people but that day, that's the death count. But still, tuberculosis is second. That's why this is pandemic. So this is the quantitative measure that why we will tell a certain disease, viral disease is pandemic. The source is our world in data. So this is the data from the Johns Hopkins site that I have collected this on June 13th, just 2 days ago. So this is the global death on June 13th, and this amount is the global infection, number of infected people, and this is the death. So global death rate is 5.53%, and this is the quantitative measure from India. So that, and the death in India is 2.87%. Still, it is so compared to the rest of the world, it is like half. So what is this virus? You know, so if you want to know the virus, we want to know what family this virus is coming from. So this virus belongs to the Coronaviridae family of virus. So what is Coronaviridae? So Coronaviridae family, this is the subfamily, this is the genus. There are alpha coronavirus that infects pigs, beta coronavirus that infects human, that is previous SARS-CoV-1 and SARS-CoV-2 both belongs to this beta coronavirus. And there is also gamma coronavirus, that is avian virus, that birds get it. So this is the phylogenetic relationship of SARS-CoV-2 from human, pangolins, and bat. You know, these two animals are very much known to you by this time, I hope. So this SARS-CoV-2 is just in between pangolin and bat. So it is more in terms of phylogenesis or genetic sequence, it is more close to pangolin than bat. But yet, the genome is different than pangolin, also different from bat. And this is the phylogenetic analysis of this virus. So this is the SARS-CoV-2 related lineage. This is the SARS-CoV-1 related lineage. And for both, you know, bat played a big role in hosting the virus. So now let's come to the detection. So what is the detection strategy of this virus? That virus testing. So you collect the sample from the nasopharyngeal swab. You collect by swabbing this, and then put it into a transport solution. And then transport this to the lab. And in the lab, immediately they lyse and inactivate the virus. And then this lyse virus can directly go to the test, or the better way is you extract the RNA by bead column-based extraction method. So you run this through the RNA binding column, the genetic material of the virus will bound to the RNA matrix, and then you elute the RNA, and then you purify the RNA and then amplify and detect it. So how you detect this? So you detect the RNA, this is the genetic material is RNA. So you add reverse transcriptase. So what reverse transcriptase will do? So it will make a copy of the RNA and make a cDNA copy. And then you followed by PCR amplification of the cDNA copy by specific primer that is from the viral sequence. So this specific primer is coupled with the fluorescent tag. So this is the Tacman primer. So fluorescent tag, and then you amplify it, and then you measure the amplification here by measuring the fluorescence. So what is the basis of this amplification? So if the number of cycle number. Here, the cycle number is more to the Can you hear me? It is why it
Yes,
is.
yes. Now you're audible.
So I was not audible before.
Just you can go back to the previous slide and describe once more.
So, okay. So
Yes,
this slide I was not audible, right?
yes, sir. Yes, sir.
Okay, okay. So what you are looking at, the color contrasted electron microscopic image of human lung cells that's infected with SARS-CoV-2. So the yellow one is SARS-CoV-2 virus, that yellow spot, each virus particle, one virion particle. And this is a human lung cell. And each virus is about 100 nanometer size. So the picture source is National Institute of Allergy and Infectious Disease. So pretty stunning picture. So this is the genomic organization of SARS-CoV-2, where we are looking at the genomic material. So the genomic material is about 30,000 nucleotide long, excuse me. So this is ORF1, ORFB. So there is a ribosomal friendship site here. This is the spike protein, and this is the EMN protein. So this is the nucleocapsid protein, E and M protein. These all proteins are structural protein of the virus. This E, M, N, and S protein. However, when the first time the virus enters the cell, it presents the genomic material or RNA to the cell, and cell has protein synthesis machinery called ribosome. So that ribosome first it translates the ORF1 and B, ORF1A and B, and make a huge polyprotein. So this big protein is non-functional until and unless it get cleaved by two viral proteases called 3CL Pro and PL2 Pro. So these viral protease chops this big polyprotein and make lot of small protein, and each protein has a crucial role in viral life cycle, including the heart of the virus, that is RNA dependent RNA polymerase, that is also coming from one of these pieces. And let's go to the next slide. So that is more explanatory. So this is the genome length, which is about 30,000 RNA, 30,000 base nucleotide. So this is what if 1AB protein. This is the polyprotein, and it chopped to different non-structural protein. That's why it is NSP. So all these little pieces do not get packaged into the mature virus. Only virus is making them inside the infected cell, but do not package this protein. But this protein plays an enormous role in viral pathogenicity and life cycle. This NSP12, one of the protein that is coming from this, upon chopping this ARF1AB protein, is RNA-dependent RNA polymerase. So this is the heart of the virus. Many drugs that is targeting this protein, NSP12, that is RNA-dependent RNA polymerase. Let's move to spike protein. So what is spike protein? None of these is structural protein. Let's not forget it. But structural protein is spike protein, E protein, M protein, N protein. These four proteins are structural protein. So spike protein is a glycoprotein, and it actually forms the characteristic spikes of the virus that we all see it in all cartoons of this virus. That which is the largest structure of the virus and essential for entry into the host cell. All vaccine currently is targeting this spike protein, against this spike protein. So E protein is the envelope protein. So this protein also functions as an ion channel. So not required for viral replication, but plays an enormous role in pathogenesis of the virus. That how virus causes pathogenesis of disease. Another protein is membrane matrix protein. So this is also present in the membrane of the virus. And nucleocapsid N protein. So this nucleocapsid N protein, it binds the genomic material. It wraps the RNA, the genomic RNA, and protects it. So now this is a busy slide, but a very important slide to understand the life cycle of the virus. So when virus enters human cell, so the receptor of the cell is ACE2. So ACE2 is angiotensin converting enzyme. So it has a totally different role in cell, that it regulates blood pressure. So, and ACE2 with the help of another protein called protease called TMPRSS2. Both are membrane bound protein. These two proteins together help to get the virus inside. So only ACE2 cannot serve as a receptor. It also needs the help of TMPRSS2. So once the virus gets inside the cell, so the host membrane was fused and ruptured, and the genetic material RNA is released inside the cell. So then first job of the virus is to dictate the host cell that, okay, translate me. So host ribosome is sort of naive. It doesn't differentiate between viral RNA and host RNA. Host ribosome see this RNA and it will keep translating. So, and then first product is this huge ORF1AB protein being made. So you mind it that this polyprotein is not functional until and unless the viral protease 3CL Pro, you can see the scissor here, the viral protease cuts it. And one of the piece is actually RNA dependent RNA polymerase or RDRP. So all of these proteins are non-structured proteins. And many of the proteins' job is to confuse the cellular immune system so that it can evade the immune machinery of the cell. So let's come back to the RDRP. So RNA dependent RNA polymerase, one of the protein, NS, one of the non-structured protein. So it transcribes again, it reads the whole genome. So transcription of the full length minus strand, this replication complex RDRP, and that synthesize again the minus strand. So from plus strand genomic material to minus strand by RDRP, and then again it synthesizes the plus strand. Plus strand, you have to understand that plus strand is the message sent strand. So the messenger RNA can be made from the plus strand. So once the plus strand being made by couple of read through by RDRP, then messenger RNA, each messenger RNA will be made, S, E, M, N, and all other accessory proteins. So when messenger RNAs are being made, so cellular ribosome will translate it and then make all viral protein. So, but these viral proteins, some of the viral proteins are membrane bound. So, virus will exploit the secretory pathway of the cell. That means the endoplasmic reticulum and Golgi pathway. So, these membrane bound protein will go through that pathway, and then that's why they get arranged into the get the proper place into the membrane through this Golgi pathway, and then the mature virus will be released by from the membrane. So it can release, or also if the viral load is high, it will just burst out the cell. So this is called lysis of the cell. And when these viruses are released, they will snatch a part of the cellular membrane that encapsulated with the lipid. Okay, so that's why you know that the SARS-CoV-2, why you need to wash your hand by soap very frequently, because this is encapsulated by lipid. Soap is a detergent, so it will break the lipid and the virus will be dead. So even if you are bringing a couple of virus in your hand, if you wash with soap, then you are safe. So, and the newly packaged virion is ready to infect another host cell. That's how. So it's a classical virus, how virus replicates and infects another cell. So now the question is, why SARS-CoV-2 is highly infectious? This is highly infectious, right? So why? So in order to understand that mechanism, why it is highly infectious, so you have to understand the mechanism, how it getting entered into the cell. So the spike protein has two subunits, S1 subunit of the spike protein and S2 subunit. S1 subunit is the receptor binding domain. That means if you consider a S2 receptor a lock of the cell, and this is the key. This is a very efficient key. It will immediately open the lock, and then the virus will enter the cell. And S2 has a different role in the cell, but virus just using the S2 receptor. So this is the angiotensin converting enzyme. So plays a very important role in renin angiotensin system. So that's why you will see that high blood pressure has a very prone. People with high blood pressure may be more prone to this infection. But also there are some people believe that S2 angiotensin converting enzyme inhibitors that for the high blood pressure medicine that many people are taking, lisinopril or this kind of medicine, catropil. So these medicines reduce the number of S2 and maybe they are protective. So, but there is no solid evidence for that. Now, if you compare the affinity of the S protein of SARS-CoV-2 and MERS. So SARS-CoV is the previous SARS and MERS is the Middle East Respiratory Syndrome. Both SARS-CoV and MERS are less infectious but highly pathogenic. Why it is less infectious? Why SARS-CoV-2 is more infectious? To get the answer, you have to know it entirely depends on affinity. So this is a paper. Can you hear me?
Yes, sir.
Yes, sir. Yes.
Okay.
Yes.
So this is a paper published in Cell that is telling about affinity, May 14th. So just 1 month ago. So they actually measured the recombinant S protein of SARS-CoV and S2 interaction. What is the affinity of this interaction of SARS-CoV-2? Hello?
Yes, sir. You are audible.
Okay. So if you look at the affinity constant, so this is the high affinity constant, that KD value. So this is actually telling that affinity of recombinant S protein of SARS-CoV-2 to S2 is the highest among S and Mars S. So this is a pretty convincing data. You know that SARS-CoV-2 targets multiple organ and cause multiple organ failure for some patients. So why it is infecting multiple organ? Because ACE2 is such a ubiquitously expressed receptor that in nasal cell is the goblet cell, high expression of ACE2. Lung cell, that secretory basal multiciliated cell, also expresses ACE2. Even the enteric cell of our enterocyte, that gastrointestinal system, that epithelial enterocyte, that cell also expresses ACE2. So even heart, myocyte, also a target of this virus. Eye, this virus can infect through eye also. It is because corneal epithelium has a significant number of ACE2 receptors. It can infect liver, bladder, kidney, pancreas, brain, even brain, even placenta, because placental cells are also target of this virus. So should I join again? Because
Yes, sir. Only a few seconds left.
okay,
Yes, we will join immediately after this session expire.
okay,
Can you stop here now, sir?
yes, I'm
Okay.
stopping here.
Okay, we'll start from this slide onward.
Okay.
Okay, I'm ending.